Good afternoon, everybody. Welcome back. Today we're going to start two sessions, that's this week, about the different kinds of infections that we place viruses into, acute infections and persistent infections. So today, acute infections. And what is an acute infection? So here is a series of graphs that explain acute and persistent infections for you. And acute infections are at the top. So what this graph is virus production on the y-axis and time on the x-axis. And the blue line is virus production. These are infection of hosts, people or some other kind of animal. So the blue line is virus, and the red bar is the disease period. So what we see at the top are acute infections, where you have a rapid onset of virus production, you have disease, not always disease, but sometimes you have disease accompanying reproduction, and then the infection is resolved, usually by immune responses, but unfortunately in the case of some acute infections like SARS-CoV-2, death occurs. So they're not always a happy ending, unfortunately. So that's an acute infection: rhinovirus, rotavirus, influenza virus. And below are all persistent infections that we're going to talk about on Wednesday. These are, my definition is infections that last the lifetime of the host. You get it at some point and you never get rid of it, and you have, in some cases, occurring bouts of disease, or you have consistently high production of virus or intermittent. We're going to cover those next time. Today we're going to talk about acute infections. And so here are the characteristics. Rapid onset of virus reproduction in the host. Limited, but not necessarily short. So short is not really the defining feature or a defining feature. It's just limited but possibly severe course of disease, production of large numbers of virus particles so the infection is transmitted to someone else, and eventually immune clearance. I didn't want to put death there because that's kind of depressing, but immune clearance or death. So these are rapid onsets and some kind of clearance. So here's the course of a typical acute infection. Again, the characteristics being rapid and self-limited, but they're not necessarily short. They're just self-limiting. So we have on this graph virus reproduction on the y-axis again, and on the x-axis it's the time. And we have initiation of infection there at the origin. And then the red line is virus production. So you see you get infected, the virus begins to multiply in you, and at some point you activate the innate defenses, which we've talked about. If the innate defenses do not clear the infection, and many are cleared by innate defenses very well, but if they're not, you reach a threshold where you then activate the adaptive responses. Adaptive responses kick in, often too late to actually clear the infection. But as a consequence of the adaptive responses, you have memory. And then if you encounter the pathogen again, you have a rapid response that's more likely to prevent severe disease. So memory is a key part here. And if you look at many of these acute infections, you see that antibody responses, at least, begin to rise just as the virus is being cleared. So it's not always the case that the immune response is clearing the infection. It probably plays a role. A very common feature of acute infections are what we call inapparent acute infections. These are successful infections. What do I mean by successful? The virus gets in you, it enters cells, it reproduces, and you shed virus. That's a successful infection. But you don't have any symptoms. Stuff you feel, that is. There's no disease. So you don't feel anything. There's no apparent disease. You don't get rash. You don't get paralysis. But you do make sufficient virus particles to spread in the human population. Now, how do we know this? Well, we do serological surveys, and we see that many people have antibodies to a given virus, but they don't remember ever being infected because they have no symptoms. So examples are poliovirus, where over 90% of infections are inapparent. They have no symptoms associated with them. SARS-CoV-2 is another one. Maybe 20% of infections, depending on the study, maybe slightly more, are inapparent. In other words, no symptoms. We do have signs, right? There are signs of infection. A sign is something that you can measure. You can take blood or some other fluid from these patients and measure virus. You will find virus. You can measure immune responses. You can find antibodies, for example. There are signs. The virus is multiplying. The immune system is engaged, but there are no symptoms of infection. And these are common public health problems, these acute viral infections. They affect millions of people each year. SARS-CoV-2, of course, is unusual. It just started. It's a brand new virus that's entered people, caused a pandemic, but it's going to be with us forever, and it's going to be causing many, many infections every year, as does influenza virus and norovirus, rhinoviruses, and many, many other viruses. And they're difficult problems because by the time you feel ill, so feeling ill is a symptom, right? By the time you feel ill, the infection may be almost over, and it probably has already spread to someone else. And this is certainly the case with SARS-CoV-2, that you have asymptomatic transmission at a high rate. People feel quite fine. They walk around, they transmit to others. And it's sometimes 20% of the time they never get sick. So they're always mixing with other people and spreading the infection. And only when they do get symptoms, if ever, do they stay home perhaps. But not everyone does that. All right, first question. Which of the following do acute infections and incubation periods have in common? Incubation period until you have symptoms. The virus is not replicating. No symptoms are visible. Immune defenses are engaged. The immune system does not respond. All of the above. Which do acute infections and incubation periods have in common? So it's number C. Immune defenses are engaged. That's what they have in common. Everything else is wrong. Let's go through it. The virus is not replicating. That can't be right. None of you picked it. Thank goodness, right? A lot of you picked just number B. No symptoms are visible. But an acute infection, at some point, you may have symptoms. So to say no symptoms are visible is not correct. Sometimes they're not if it's an asymptomatic infection. Certainly that's the case for an incubation period. I totally agree. But there are some points in an acute infection when symptoms are visible. So that's not right. And you can only pick one, so immune defenses are engaged in an incubation period and an acute infection. Both of those are correct. It's a tough question. I agree. Even I had to think about it a bit. And I made it up. So today we're going to talk about 5 viruses that cause acute infections. I don't have SARS-CoV-2 here. It also causes an acute infection because that is going to be later in the course. We're going to have a separate lecture about 2 pandemics, HIV/AIDS and COVID-19, in one lecture. Today, influenza virus, poliovirus, Measles virus, Norovirus, and West Nile. Why did I pick these 5? Because they represent different modes of transmission. Influenza virus and Measles virus are respiratory transmitted, poliovirus and norovirus fecal-oral, and West Nile by mosquitoes. And there are also different kinds of viruses as well. Let's start with influenza. Anybody have influenza this year? You don't have to admit it, I understand. Did anyone? I did not. You had influenza. Did you have a vaccine? Yeah. Oh. But you didn't die, right? No. Clearly. So it prevented you from getting really sick. You stayed out of the hospital. I'm sorry to probe your medical records. You don't have to answer. But it's in the interest of academics. I did not. I had a flu vaccine. I haven't had flu for years because I worked on flu as a graduate student. I probably have the highest antibody titers of anyone in the world. So there are 3 types of influenza virus. Remember, this is an envelope virus with a segmented negative stranded RNA genome. 3 types. Type is an interesting word. These are different influenza viruses. If you get infected with a type A, you're not going to be protected against B or C. That's one of the differences. And A and B cause a similar disease, a respiratory infection that we'll talk about. C, mostly inapparent or mild upper respiratory tract illness, common cold. That's another name for a common cold. Only influenza A virus causes pandemics. Does anyone know when the last influenza pandemic was, by any chance? 2000 and what? I heard the right number. Yeah, 2009, yes. 2009, the so-called swine flu pandemic. And the hallmark of influenza is antigenic variation, which we'll touch on today, and we're going to return to when we talk about evolution. Very interesting. So the virus is transmitted from person to person by droplets produced by coughing, sneezing, and talking, which we have discussed before. There are droplets of different sizes that go different distances. Probably mostly the large and the intermediate-sized droplets. Direct contact with an infected individual. What does that mean? Well, just touching someone in some way, even just shaking hands, and they've just touch their nose, you shake your hands, and then at some point you touch your nose, you introduce virus to your nose. We do that all the time. We're always touching our faces. In fact, you can avoid most infections if you keep your hands away from your face until you can wash them. So if you spend the day out and about, you're in the subway particularly, try to keep the hands away until, I have to scratch my eye now, so I can't keep my hand away from my face, until you can wash your hands. Or you can carry the hand sanitizer with you. Direct contact with individuals or surfaces. There's some evidence that contaminated materials remain infectious for some time and you could acquire infection that way. That's not the case with SARS-CoV-2. Don't think, mostly it's transmitted by droplets. The virus, you inhale it, so you either inhale it or it enters your upper tract, the portal of entry of these respiratory viruses. So it enters the upper tract through your mouth or your nose and reproduces in the mucosal epithelium of your nasopharynx, this upper part of your tract here, lined with epithelial cells. It has a nice mucus layer to protect you. It has a basement membrane, of course, but in some cases the virus will attach and start to reproduce in those epithelial cells. The basement membrane remains intact. This virus does not get into your circulation. It does not become systemic. In most people, the virus goes down into the trachea. And so sometimes when you have influenza, you can have a burning sensation under your sternum, which is roughly where your trachea is. And some people, it will also go down into the alveoli and cause pneumonia, which is a more serious disease, typically in elderly patients. Now, if you may remember from lecture, no, you don't remember this, but I'll tell you, from lecture number 5, virus entry, the receptors for influenza viruses are sialic acids. They're sugars attached to proteins. And human and avian influenza viruses attach to different sialic acids. Human viruses mainly alpha-2,6, avian alpha-2,3. Avian influenza viruses like H5N1. Did you hear they just culled all the pheasants and chickens on a farm up in Ithaca because they got infected with avian H5N1? Too bad. You know, there's the little chicks that they give to schools so the kids can, I don't know what they do. They look at them and say, this is science. But they had to kill them all. It's so sad because it's hard. So the wild birds flying over infects these farms. So avian viruses bind alpha-2,3. They don't infect people very often. There have been fewer than 1,000 H5N1 avian influenza virus infections since we started tracking it. About 800 deaths, maybe fewer. And part of the problem is that alpha-2,3 sialic acids are mostly in our lower tract. So here you see the bronchioles all the way down in the lung. They have alpha-2,6 and 3. The bronchus has 2,3. The trachea has 2,3. So the virus has to get an avian influenza virus has to go down pretty deeply into the tract in order to infect us. So it's rare. However, and you know, the alpha-2,6, which is the human targeting sialic acids, the alpha-2,6, which bind human, are all the way up throughout the tract. However, there's one. So people who get avian influenza, H5N1, typically sleep with chickens because they farm chickens and they only have one house, and the chickens live in the same house, and so they're inhaling deeply all night and they get infected. But there's another way humans can get infected. Our eyes here, our cornea, our conjunctival membrane, the nasolacrimal duct, they all have alpha-2,3 sialic acids. And so it has been documented on chicken farms that you can have chicken farmers get infected. It starts in the eye, and then the virus drains through the nasolacrimal duct, right? That takes your tears from the eyes into your nose, and then it can get to the rest of the respiratory tract. So not as hard as we thought it would be, but still a very rare infection in humans. So we are worried that H5N1 is going to cause a pandemic in people. We'll talk about that later. But so far it hasn't. It hasn't even spread extensively in people. Uncomplicated influenza, short incubation period, onset of headache, chills, dry cough, flu-like symptoms, high fevers, myalgias, malaise, anorexias. Your fever peaks within a day, and then it goes down, but then you get more respiratory symptoms. You get a productive cough. What does that mean? You're coughing up mucus, and this can last for 1 to 2 weeks. Does that sound familiar? Yeah. So it's a long infection, and you often have pain substernal pain because the virus and the immune system is destroying cells in your respiratory tract. So that's uncomplicated influenza. We diagnose it in a couple of ways. Influenza-like illness, fever of 100 degrees, cough or sore throat. That's it. If you go to student health and you have a fever of 100, cough or sore throat, they'll say you have flu. Sometimes they will test you. There are rapid lab tests, but they're not so accurate. They may just give you antivirals right away or tell you to suck it up probably and go take some over-the-counter stuff to make you feel better. And of course, if you have no other known cause of illness, if you haven't been tested for anything else. So there's a rapid antigen test that you could do in a doctor's office. You take a nose swab and you put it on the test. There's also PCR, viral culture, serology, but they take more time, of course, and have to have a specialized lab to do that. But in a flu season, they will just say you have flu without even testing many times. So here are 2 experiments that show you the relationship between shedding and symptoms. These are actually challenge experiments. We can do challenge experiments with certain strains of influenza virus, especially in people who are vaccinated. What does a challenge experiment? It means you give people virus in their nose, which we usually don't do for most viruses. But this is one case we can take a strain that's mild, and we use people who are immunized. So on the left, we have inoculated people. You pipette the virus in their nose. They make you sit back in a chair, and they pipette it in your nose, and then sometimes they put a clothespin on your nose so it doesn't drip out for like 10 minutes. It's really similar to the real-life situation. That's how you get flu, You walk around with a clothespin. I'm joking because I think it's crazy that you have this clothespin because it's not the way it happens, but that's what they do. And then on the left, we are looking at virus titers. They take nasal washes at different times, the x-axis, and we're looking at virus shedding and then a symptom score. A symptom score is they ask you every day, how much have you blown your nose? How much does your head hurt? All these things, and they add up and make a score. So you can see the viral shedding. It's sort of beginning before symptoms are very accentuated. These are pretty mild symptoms. So here you have that first time point. You have a lot of virus shedding. So you have about a day of shedding before you really feel lousy. And so that could infect other people. And then the shedding peaks at, look at this, 2 days. This is really rapid. And then goes down, and by 8 or 9 days, no more virus. All right, so that's one. And then this is a second challenge study on the right where we're looking at more parameters. Again, on the left we have antibody. Actually, we have multiple y-axes. We have antibody titer, we have virus titer, and the nasopharyngeal wash. And then on the right Y, we have interferon titer in the nasopharyngeal wash. So you take a little bit of PBS saline, you put it in the nose, and then you measure these things. Yeah.
Why was it considered unethical to not for influenza?
So in the UK, they didn't think it was unethical. They did it. It was approved by the authorities. I thought it was unethical because there's so many unknowns about SARS-CoV-2, including what fraction of people get long COVID, right? Symptoms that last for many years. We don't know how many years, but a lot of years. And so even though they picked young, healthy people, one of them lost his sense of smell, and I don't know if they've recovered it yet. So, I mean, I guess they were willing, these risks are explained to you, but it also could die. You don't know. That's the problem. Even if you're young, you may have some underlying condition you don't know about, and you could die. Whereas with influenza, we've studied for so long, we've done lots of challenge studies. We know that with a mild strain and also with vaccinated people, it's not going to be a problem. But I still think long COVID, I mean, right now, today, people say to me, the only thing I'm worried about getting COVID is long COVID, right? That's what people are still worried about. And so I don't know how you can do a challenge study when that's really the prime worry. Anyway, on this right-hand graph, we see virus peaking again, 2 to 3 days. Interferon goes up right away, which is what it's supposed to do. We don't have any symptoms here, but these are all antibody. Look, the antibodies are hardly getting going when the virus is gone at day 6. There's a little bit of antibody. But it's mostly interferon that I think is taking care of this infection. And then you have antibodies rising much later, and then, of course, you get memory for that. But again, you don't know that from this, but you are shedding before a symptom onset. So we track influenza hugely in the world. We'd like to know where it is, and this is an example of one of the trackings over time. So there are many, many laboratories in many countries that will get samples from people, see if they're influenza, and do some studies on them. So here, for example, are influenza numbers of isolates here on the left and percent positive on the right. And so these are different years. So you can see the seasonality of influenza. First of all, here's 2004, 2005. You can see the numbers start going up. So the line is the total number of isolates, and then the bar graphs are the percent positive. As you can see, it starts to go up in week 50-ish. That's the end of the year, right? 52 weeks in a year. And then the peak is like week 8, which is February-ish, and then it starts to go down by week 18. More or less similar every year. So winter disease in the Northeast, this is U.S. And also these different colored bars are the different strains of influenza virus that can circulate. So we have influenza A and we have influenza B. And you can see the B is in green. It was particularly high in 2007 and 8. And in some years, we have more H3N2, and in other years, we have more H1N1, 2 different subtypes. And they vary according to the year. So that's an example of seasonality. This is the burden of influenza in the U.S. Typical year, between 9 and 45 million illnesses in the U.S. It's a lot. And these are mostly from the top range is from the 2017-18, which is a particularly severe season. Then 140,000 to 800,000 hospitalizations, and then 12,000 to 61,000 deaths. So flu is serious. It can kill you, and that's why you need to be vaccinated. And here on the right are the same triangle for different years. So you can see some years, 2011, not a lot of cases. But there's always a similar burden of deaths, which is the very top of the triangle. So that, because there are so many deaths, that's why we vaccinate against influenza virus. And here's some more data. So as I said, we track it really intensively. So I want on the left the 2 graphs on the left. That on the top is 2019-2020 season, right, just before the pandemic. And then on the bottom is 2022-23. And again, these are the different subtypes of influenza by week. And I just wanted to show you that on the top, what is prevailing, I can't see it because it's out of focus. The brown is the H1N1, the 2009 pandemic strain. And on the bottom, the predominant strain is H3N2. So it depends on the year. They kind of seesaw back and forth. But again, you can see the peak is more or less in February-ish. top right is pneumonia and influenza mortality. So for many years, we tracked people who died of pneumonia and influenza as a way of gauging how many people died of flu. Because when you die of influenza, they don't always put influenza on your death certificate. They put pneumonia, for example. Some physicians will do both, but that's why we track both pneumonia and influenza, because the assumption is the majority of pneumonia deaths in winter are caused by influenza. And so you can see this cycle every year, peaks in the winter and goes down. The two black lines are what you would predict as the norm for influenza and pneumonia deaths every year. And the red is the actual number of influenza and pneumonia deaths. And you can see in some years, 2017 and 18, we have way more deaths than we would have predicted. So it's a particularly severe year. And finally, down here, this is a map of cases per state. Every state reports its influenza cases to the CDC, and they compile this. So this is actually a movie I'm going to play for you. It's the season of 2022, 2023. It's starting in week 40 at the end of 2022, and then it's going to go into 2023. This is very exciting. Look carefully, okay? You're going to just love this. So you can see as the time goes, we're going week 44, 45. We're getting more and more cases because the red is a lot of cases. Week 49, 50. Now it's starting to go down at the end of the year. Already week 1, 2. This is a very early end of the influenza season in the U.S. So February 2023 and now March, which is where we are now. Isn't that exciting? You want to see it over and over. You could get it from the CDC website. It's not as good as the plaque movie, but it's pretty good. Anyway, we track on a very granular level. Finally, one more. Since COVID, we have now added pneumonia, influenza, and COVID mortality to this, because COVID is a new respiratory disease, and so we do all three. And again, here's the epidemic threshold, the two black lines, the high and the low that we would expect every year, and the red is what we actually observed. Here's 2019. And then, of course, all hell broke loose in 2020. Our influenza, pneumonia, COVID death went really high, as you can see. So the red is percent deaths to pneumonia, influenza, and COVID. And the blue is the COVID-coded deaths. So on the death certificate, it says COVID. And you can see that in some cases, COVID is accounting for most of the PIC deaths, so not a lot of influenza. And in some cases, there was a peak, which probably is a lot of influenza. But, influenza during the pandemic years was suppressed because of the masking and isolation that we were doing. And if you look at flu diagnoses, I don't have any for you because there are hardly any cases diagnosed. And one more, this is the percentage of outpatient visits for respiratory illness. There is a network, the U.S. Outpatient Influenza-like Illness Surveillance Network, ILI-NET, and this is, again, another metric reflecting the flu season, people going to outpatient visits for a respiratory illness. It's not all influenza, but a good amount of it is. And you can see peaks in different times of the year. Here's, again, the end of the year, week 52, and then the next year here. And so our current season is 2022, 23, these red triangles. This peaked, as I said, very early on. And you can see in other years, it tends to peak later. And then it's over by, week 12 or so, January, February, March, April-ish. It's all over. Okay, so that's influenza. Complications occur. You can get pneumonia. As I say, the virus goes down into the lung and makes a lot of damage. You can get secondary bacterial pneumonia. This means you're starting to recover from influenza, and then suddenly the bacteria invade and cause a serious pneumonia. Muscle pain. You can have heart involvement. Remember, your lung capacity is compromised. Your heart is working harder, and in some people that causes issues. And then something called Reye's syndrome, encephalopathy, and liver damage. The etiology of that is unknown. We have over-the-counter stuff you can take to make you feel better. We have antiviral drugs, Tamiflu, Relenza, Flumidine, Xofluza. We'll talk about those when we talk about antivirals. But you have to take those within 1 or 2 days of symptom onset. When you first feel that scratchiness in your throat, you got to go get a prescription. If you wait too long, it's of zero use. But if you take them within a day or two, they will help shorten the course, which is already quite short. Right? And we have vaccines, and we'll talk all about those later. Next question. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. Incubation period 1 to 5 days. Fever peaks within 24 hours. Coughing and weakness can last for 2 weeks. All of the above. How'd we do? Most of you got all of the above. Very good. Only one person, I think. That's what that 4% is. One person got transmission may occur. That's true, but they're all true. 1 to 5 days. Fever peaks in 24 hours. Coughing and weakness can last for 2 weeks. All right. Measles. I had measles as a kid because they didn't have a vaccine back then. But most of you should not have had measles. You should have been vaccinated. This is another envelope virus with a negative strand genome. It's a member of the paramyxovirus family with an enveloped negative stranded helical nucleocapsid. There's an electron micrograph on the left showing the lovely nucleocapsid. One of the most contagious human viruses. I debated whether leaving the R naught here was a good idea because, as I told you earlier, R naughts are all differently calculated. And however, this one is always consistently high. So 15 means that one infected person could infect 15 others. So if I had measles, most of you would probably be infected, including the ones you in the back row, because this virus goes on those tiniest droplets that go very, very far. But I don't have measles, so don't worry about it. Pathogenesis, one serotype, which means not antigenic variation, not multiple types like influenza. Infection gives you lifelong protection. It's transmitted by these droplets and aerosols. And you are maximally contagious 2 to 3 days before you know you're infected, before the rash appears, where you say, it's measles, the kid has to stay home today. So asymptomatic transmission. But there's very little asymptomatic disease. There's pre-symptomatic transmission, but very little asymptomatic disease. Pathogenesis, quite interesting. So you inhale the virus. Droplet inhalation reproduces in the epithelium of the respiratory tract, not actually in the epithelial cells. It attaches to dendritic cells and macrophages, which bring it through the epithelium, makes its way to lymph nodes, and reproduces in lymphocytes. So it's not reproducing in the epithelium, but the DCs and the macrophages bring it in. They go to the lymph node to try and see if it's foreign, and they bring it in there, and it multiplies. You have a primary viremia, virus in the blood, that spreads it to many other organs. The skin is one, of course. The rash is typical. But it can also get in the brain. About 1 in 1,000 kids with measles have encephalitis. And then it has to get out again, right? So now it's in the blood. How does it get out to transmit to someone else by the respiratory route? Really interesting. So it's in the blood. And in the blood, it can then reach the basolateral surface of the respiratory epithelium. That's where it needs to go to transmit. On the basolateral surface, there's a receptor called Nectin-4. The virus, the virus is that. So these are white blood cells bringing the virus to the basolateral domain. The virus are these tiny particles attaching to Nectin-4. They enter the epithelial cells, they reproduce, and they're released apically so they can get into your lumen of the respiratory tract and be transmitted. Isn't that amazing? So this virus has to go to other hosts. It has to go to another host to endure, right? And the way it does so from the blood is to reproduce from the bottom of the respiratory epithelium and out. So on the apical domain of the respiratory epithelium, there's no Nectin-4. So the virus cannot enter in the beginning of infection. That's why the DCs and macrophages are picking it up. Very interesting pathogenesis. So there's a child with measles, rash, all over the body, from the face to the extremities, high fever, runny nose, cough, conjunctivitis in your eye, Koplik spots What's a complex spot? Well, in the days when we had a lot of measles, you could look in the mouth of a kid with measles before they had a rash. So let's say they're going to school and they have a fever. They don't feel well. They're complaining. They go to the school nurse, and the school nurse could look in the mouth, and it's not very visible here, but there are these white spots that are called Koplik spots, and only measles does that. So you could say, you have measles, go home. You got to call mom or dad to go pick you up, and et cetera, et cetera. And what are they? The virus is reproducing in the oral mucosa, and it's causing the cells to fuse. That's what this virus does. It makes syncytia, and those fusing give you the white spots, the Koplik spots. So there's a timeline of measles infection. So we have an incubation period where the virus is multiplying. Then there's a prodrome where you have a lot of symptoms not characteristic of the disease. You have fever, you have virus shedding, Koplik spots, and then the rash appears at around day 14. So, a little longer than we've been talking about so far. This should be antibody. That's an error in the textbook, which we will hopefully correct in the next edition. And then recovery. And by, the antibody plays very little role in recovery from infection, but now we have memory that's going to protect us against the next one. As I said, protection is lifelong. There are serious complications with measles. And this is why kids need to be vaccinated. 1 in 1,000 kids get encephalitis. Never a good thing to have virus reproducing in your brain. You can get bronchitis, pneumonia, you can get ear infections, and it will kill some kids. 1 to 2 in 1,000 kids will die of measles. That goes up to 28% in countries where the kids are malnourished. So even more severe causes, a good reason to be vaccinating kids everywhere. Next time we're going to talk about a long-term sequelae of measles, subacute sclerosing panencephalitis. Very rare sequelae, but fatal. And, of course, immunosuppression, which we talked about last time, which leads to other infections, and that's the main cause of death in kids who are malnourished. So we introduced the vaccine in the 60s because before the vaccine, there were a lot of cases of measles. This is just in the U.S., 3 to 4 million a year, 400 to 500 deaths, 48,000 hospitalizations, and 1,000 kids every year had chronic disability from encephalitis. This is what a virus reproducing in your brain can do. So measles vaccine was introduced in the 60s, and it stopped endemic transmission of the virus by 2000. And it's part now of a trivalent vaccine called measles, mumps, rubella, MMR. Unfortunately, in 1998, the gastroenterologist in the UK, Andrew Wakefield, a gastroenterologist working on a virus, he said measles vaccine, MMR, causes autism in a paper that was completely flawed and which was eventually retracted. But that led to people saying, I don't want to immunize my kid. And so lots of outbreaks in the UK and England, and those viruses get imported to the US, and they cause outbreaks in communities that don't immunize. So Rockland County, where there was a case of polio last summer, they don't like to vaccinate, and they have outbreaks of measles regularly. So here's the case, measles cases in the US, vaccine introduced in the 60s. You see a lot of cases per 100,000. We bring it very low. We're almost at eradicating, and then the Wakefield thing happens, and we have outbreaks every now and then. Big one here in 2019, 1,200 kids, 120 last year. These are mainly in communities where parents still think MMR causes autism. It does not. It's been shown many times. Here's an outbreak in New York City, 2018. And you can see all these cases in different boroughs of Manhattan. And again, mainly in communities that do not vaccinate their kids. This is global measles. A lot of cases of measles globally. We're here at the end 2020. Not much reporting during the pandemic. 413,000 cases in 2019. They're all vaccine preventable, and a lot of kids will die as a consequence. These are the countries that have a lot of measles. The darker reds are the more measles, and we have, still a lot of measles in the U.S. These are the countries with the most measles outbreak. For last year, the top country was India with 48,000 cases, Yemen, Somalia, Ethiopia, Indonesia, etc. Again, all under-vaccinated. A lot of countries put off their measles campaigns because of the pandemic, but even pre-pandemic, a lot of countries simply didn't have the resources to vaccinate everyone. And this is a shame because all of this is vaccine preventable, of course. We'll come back to measles vaccine again later, and we'll explore it further. Our next and last question is, which of the following is a good reason to get measles vaccine? There is a 1 in 1,000 chance of acute post-infection encephalitis. There is a 1 to 2 in 1,000 chance of death from measles. Each infected person spreads measles virus to 15 others. Immunosuppression can lead to secondary infections. All of the above. So Andrew Wakefield is now in Texas. He was disbarred in the UK. They took away his MD. But he went to Texas where he can practice medicine. And he talks about why MMR causes autism and other vaccines. Of course, he said all the COVID vaccines are terrible. And you know why he does this? Because he gets paid millions of dollars to say these things. And this is an MD. You guys, some of you are going to go to medical school. You're supposed to help people. You're not supposed to hurt them. I just cannot believe that he would rather have a lot of money than to take care of kids, because the vaccine does not cause autism. It does not. If you look at the data, it's quite clear. 23. All right, what do we have here? I think that's number 2. Very good. Yes, all of these are good reasons to get measles vaccine. I put this here on purpose so that you would remember that. And, you're going to have parents who say to you, if you're a pediatrician, why should I vaccinate my kid against measles? It causes autism. So you need to sit down and take some time. You're going to be really busy, and your receptionist is going to say, Doctor, you have 10 patients waiting. You got to sit down with this patient and tell them why it's important. You cannot let you being too busy compromise their health. And you need to learn that measles vaccine is complete. It's not 100% safe. No vaccine is 100% safe, but it doesn't cause autism, that's for sure. Now, you want to talk about unsafe vaccines. One of the polio vaccines is somewhat unsafe, but the anti-vaxxers don't know it because they don't read. And they don't listen to my lectures, so they don't know it either. And we're going to talk about that next time. It's a really interesting story. But for today, poliomyelitis is the disease caused by polio. This is a naked icosahedral RNA virus with plus-stranded RNA, rather small, 30 nanometers or so in diameter. You acquire this virus by ingesting stuff that's contaminated with fecal material. And you may say, well, how can that happen? Because we don't wash our hands. We shake someone's hand, we touch something that's contaminated, we put it in our mouth. Kids are really good at doing that, but adults are pretty bad at hand hygiene too. You ingest the virus, it goes through your stomach into your intestines. That's amazing. Low pH in the stomach, high pH in the small intestine, bile salts, proteases. Polio is like, what else do you have to throw at me? It's just retaining infectivity through all that. It gets to the intestine, reproduces in the epithelium, and then you shed a lot in the feces. That's how it spreads from person to person. It sets up a viremia. It gets into the blood, so it goes through the basement membrane, gets into the blood. And then in 1% of infections, it gets into the central nervous system. It seems to go into the muscle, travel by axonal transport to the spinal cord, eventually the brain. It destroys motor neurons, and you get paralyzed. That's the pathogenesis. But, the paralysis is an accident. The virus, in order for the virus to be transmitted, it needs to stay in the intestine. Entry into the CNS doesn't get it transmitted. So that's an accident, which unfortunately has claimed many, many children and continues to do so. Here's an example of what the virus does in the spinal cord. This is from a mouse. This is a mouse that was paralyzed after infection with polio. But you can see all these dark cells. These are probably lymphocytes coming to the spinal cord and trying to clear the infection. And then the green are neurons. These are neurons infected with poliovirus. The green is we've stained it for poliovirus RNA. And so you can see they're larger cells. They're kind of pyramidal. There's one all by itself there. Axons and dendrites full of viral RNA. So eventually the virus is going to destroy that cell. And if you destroy enough neurons that innervate a muscle, then you get paralyzed. So here's the course of polio infection. This is actually a hand-drawn graph from the 1950s. You know, before we had computers, we had to draw figures by hand. And this was one of it. It's just beautiful, I think. It's a summary of many cases of polio. And so here's exposure on the y-axis. Then we have days after exposure. And on the x-axis. And then on the y-axis are different, we're looking at different things. So on the very top, we have temperature. 7-day incubation period, then we have a spike in temperature, headache, and sore throat, nausea, right? Flu-like symptoms. Most of the kids get well. And at the same time, they have a lot of virus in their feces for a long time. Look at 30 days. Virus and throat secretions as well. Serum antibody comes up later. You have a viremia, that's down here in the third panel. Okay, then in 1% of the kids, the infection continues. You have another spike in temperature. So now we're talking about 12 days, and then in the next 17 days or so, headache, nausea, and paralysis will happen there. And their virus is in the CNS. You can see from day 10, we have now a spike of virus in the CNS that correlates with paralysis. So, it's an unremarkable infection, except 1% of the kids sadly get paralyzed. And that's why we made vaccines against this virus and continue to do so. Humans are the only known reservoir of infection. No other animal seems to get it in nature. It's spread by fecal-oral contamination. In temperate climates, it peaks in the warm months of the summer. And there is a complication known as postpolio where, you can be given therapy and regain your limb functions after paralysis, depending on how severe the paralysis is. And then 30 to 40 years later, suddenly you start to lose limb function again, and this happens in about 25 to 40% of people. It's not an infectious virus. The virus is not there. You could call it long polio, I think. And what happens is you have lost a good number of neurons to infection. And as you age, you lose more neurons naturally. And so because you've lost so many to infection, you can't handle it and you lose limb function. Yes? What is the what?
Iron lung is what?
The iron lung is to allow, so in certain people, the paralysis affects the respiratory muscles. You can't breathe, so they put you in an iron lung and they use pressure to breathe for you. So the chamber, you lie in the chamber, it fills with air, it pushes your chest in, then the air goes out and your chest expands. So before we had high-flow oxygen tubes to put into people, that's what we had to do. And you could be in that for a while and then recover your muscle. And most people got out of those iron lungs, although there are still a few people in the world who are still choosing to live in an iron lung, even though they could have a nasal cannula to supply oxygen. They want to do that. Now, here's the cool thing, or interesting. It turns out when you put a tube in your lung and put 6 liters of oxygen a minute, it really screws you up. Most of those people are not the same if they survive COVID. And many physicians wished they had iron lungs because it's a much gentler way to deliver air to patients, but we don't have any of those anymore. I saw one once in a museum in Germany, and there's one on eBay too for $30,000, but it's huge. It's really huge. So this is polio in the U.S. We had many cases, up to 20, 30,000 cases of paralytic polio a year, peaking in the 50s, the baby boomers. So the World War II people came home, they had kids. That baby boomer, post-World War II generation supplied more subjects for polio, so the numbers went way up. So we made a vaccine, we made an inactivated and an oral vaccine, and we eradicated it in the U.S. I'm going to tell you this story next time. Although the case last summer was the first case of polio since 1979. And I'll tell you why that happened. And also the difference in those vaccines. Norovirus. This slide was given to me by a Stanford gastroenterologist. He said, Vincent, you have to use this. Did you know in a 24-hour period. 200 million people in the world have gastroenteritis, vomiting and diarrhea. And the amount of water they pass equals the volume of water passing over Victoria Falls in 1 minute, which is 65 million liters per minute. Next time you're at a party, you should tell people this. I think it's an impressive 1 minute. That's a lot of diarrheal virus. So what does this? It's mostly norovirus. So when you talk about foodborne outbreaks of gastroenteritis, 48 million a year in the U.S., there's the pie chart breakdown. 58% are caused by norovirus. We used to think it was all bacterial before we discovered norovirus and said, hey, 58%. You know, you have your Clostridium and Staph and Salmonella, can't be. But norovirus, and there are other RNA viruses that can do this as well. Norovirus is a calicivirus. It's a plus-strand RNA virus. And on the very first day of this course, I showed you a picture of a whale that I said was infected with a calicivirus, and that is related to norovirus. With this virus, we can do challenge studies also, because it doesn't kill you as long as you drink a lot of fluids. Kids do die of norovirus because they can't stay hydrated. But if you're hydrated, you'll be fine. So they can do challenge studies, and we know that it takes about 18 viral particles to infect you. So it doesn't take a lot, and it's easy to see how it can be contaminating you. So, by the way, challenge studies are often done with medical students. They take them on a Friday, and they feed them basically filtered diarrhea from other patients because they can't grow this very well in culture. And then you get sick and you vomit and have diarrhea, and then Monday you're well and you go back to class and they give you $300. So if you're in medical school, maybe you want to jump to do that kind of challenge study. And remember me when you do that. Anyway, we can do challenge studies with norovirus. So this is what I call a two-bucket disease, because you're vomiting and you're having diarrhea. Okay, that's a great term. It's a microbiological term. Remember it and use it. You will get laughs. Fecal-oral spread, of course, not washing your hands. The virus particles, like polio, go through your intestine. They reproduce in intestinal epithelial cells. They seem to blunt the villi. So here on the left, lower left, is normal villus. On the right, from a norovirus-infected person, they're blunted. We don't know why. We don't know why you vomit. We don't know why you have diarrhea. The basis for that is unknown. But it causes quite explosive vomiting and diarrhea, and that's, of course, how it transmits. Now, I always say this is a highly unsophisticated virus because look at poliovirus, which transmits as well as this virus. No vomiting, no diarrhea. Extremely sophisticated, and this one has to make you do all of this, which is very effective at transmitting, but it's absolutely not necessary. So transmission is fecal, oral. It can also be transmitted when you vomit. When you vomit, you do a nice aerosol, right? So if there are people around you, you can infect them. Contact with contaminated material fomites, you can get it from food, water, environmental contamination. So oysters often have it because they're sitting in beds in the ocean and filtering the sewage that we're putting out, right? Raspberries, if you wash raspberries with contaminated water, or if the food handlers are shedding, which they can do so, and they can shed asymptomatically, they can contaminate food, they can contaminate food in food preparation areas and so forth. Rapid incubation period, sudden onset of vomiting, diarrhea, stomach pain, short illness, and 30% asymptomatic infection. So that's a problem, right? You could be shedding and you go to work and you're preparing food, right? That's part of the problem. They're shedding peaks after illness onset. You can shed for 56 days, so you could shed way after you're healed. And when you go to a restaurant now, you go to the bathroom, there's a sign in every bathroom. What does it say? Employees must wash their hands. Do you know why that's there? Norovirus. It's there because of norovirus, not because of anything else. So you look at it and you can think of that next time you see it. The immunity is short-term only. You can be reinfected many times in your lifetime, and there's more than one strain for sure. The reservoirs, humans, It looks like we can infect some other animals, very much like we can infect animals with SARS-CoV-2, but the significance of that isn't clear. It affects all ages of people. Peaks in cold weather, but it happens year-round. We happen to have come off a big norovirus outbreak right here in the U.S. in the past month or so. And the outbreaks often occur in semi-closed environments like nursing homes, hospitals, cruise ships, schools, where people are together for extended periods of time, and it favors person-to-person spread. Now, what do you do right now? The only thing you can do is rehydrate, prevent dehydration. A vaccine is in development, so at some point we will have one. But there are deaths in countries where they cannot hydrate the kids. And that's unfortunate. You say, why can't they hydrate the kids? Well, they're very young and you can't give them enough water by mouth, so you have to do IV, and they can't do that. They either don't have it, or they don't have enough, or they can't afford it. So here are the breakdown of cases, first on the left by settings. And you see cruise ships there in green. It's only about 10%, although the cruise ships get a lot of publicity when they have to shut down for a norovirus infection. But the most transmission is in restaurants and catered meals, also retirement centers, nursing homes, hospitals, and schools. So that's the setting. And on the right, the mode of transmission, person to person, or foodborne or waterborne. This happens on airplanes, too. People throw up on airplanes. This was an example of a Air New Zealand flight where a sick passenger, right, they vomited into the bag, but they made a mess, and the crew cleaned up, and that crew got sick, and then on every successive flight, at least one or more crew members got sick with norovirus because it gets aerosolized in the airplane and can spread quite easily. And plus, the crew members have to clean up. I have a listener of one of my podcasts who's a flight attendant, and she says when someone throws up, she runs the other way so that other people will clean up for her. So it can be aerosolized in vomitus as well. So why are cruise ships susceptible? So here's just a list of some cruises over the years where there have been outbreaks of gastroenteritis, and they've had to come back to port, and you have to get rid of everyone. Well, you get everyone off the ship, and then you have to sterilize the whole ship, basically. It's very expensive for them, and mostly they're noroviruses. But you see, sometimes they're unknown agents. They're probably other viruses as well. So a listener of TWIV is a cruise ship agent in Norway, and he wrote me once, and he said they have installed on their ship these hand washers where you put your hands in and it automatically washes your hands for you. And he said it's really cut down on the amount of contamination they have. Very interesting. So why are they associated with cruise ships? Well, we track infections on cruise ships, so we're looking for it so that we can report them. Close living quarters, new passengers coming in. But all the people working there are tested to make sure they're not shedding. And so it's the passengers that are coming in that are doing this. How do you protect yourself? Wash your hands, rinse your fruits and vegetables, cook your shellfish. I know you like to eat raw oysters. They're cool raw, but you're taking a chance. Keep your surfaces clean. If you're sick, don't prepare food for others. And stay away from the mints when you leave the restaurant. Those mints, people have done studies. They're full of poop. They're actually full of coliform bacteria, which is a readout for fecal contamination, because people go to the bathroom, they don't wash their hands, and they grab some mints. Even if they're wrapped, don't touch them, okay? Because they have norovirus on them, and that's a common way of spreading it. So stay away from the mints. Okay, our last virus is West Nile virus. Unfortunately, not as amusing and interesting as norovirus. This virus did not come from Egypt. It came from the West Nile district of Uganda, contrary to this cartoonist who thinks that this virus came from the West Nile. It came from Uganda originally in 1937. And it was absent from the Western Hemisphere until 1999, when it first showed up here in New York City in August of 1999. Some people in Queens started getting very sick. Many of them developed encephalitis. Birds started dying. And in the Bronx Zoo, there was a very astute pathologist who said, this is unusual, and she figured out that it was a virus infection. Eventually, it was discovered to be West Nile virus. And the isolate that started in New York, its genome is identical to a virus from an Israeli goose. So they have goose farms in Israel, and there was an isolate on that farm that was identical to the isolate in New York. And so the assumption is it came over some way. A goose didn't come over. They don't come that far for sure, but it may have infected a person who came over and so forth. Or actually, that's not possible, sorry. People don't transmit it to each other. It's only by mosquitoes. When you're infected, a mosquito infects you, and you don't have enough virus in your blood for another mosquito to bite you and transmit it to another human. So the only way that could have happened is for a mosquito to have come in the airplane, which happens. Anyway, it's spread throughout the North and South America. Now it's throughout the U.S. and South America. And it infects many birds, it infects many mosquitoes and many other vertebrates as well. So it's here to stay. The transmission cycle, the natural transmission cycle is between birds. And mosquitoes transmit the infection from one bird to another. And these birds have had the infection for many, many years, so they don't get sick. Occasionally, the mosquitoes, of course, will bite other hosts, humans or horses, deliver the virus to them, and then the humans or the horses can get sick. But as I said, the humans and horses cannot transmit infection via mosquito to someone else because they don't have enough virus in them. And so in the U.S. outbreak, what happened was the mosquitoes started biting birds that never saw the virus before. And, at the Bronx Zoo, those are like the exotic birds, and they died. But even the crows and the blue jays here in the U.S., which are natural hosts in other countries, they died because they'd never seen the virus. They had no immunity. It's like introducing a virus into the new world by explorers, right? It's the same thing. You have a completely naive population. So it's transmitted by mosquitoes, as I said. So again, the mosquito has taken blood meals on a bird, and then it's accidentally infecting a human. Incubation period. 20 to 30% of people develop a flu-like illness. It's called West Nile fever. Can't tell that it's any different from any other virus infection. 80% of people have no symptoms whatsoever. So a lot of asymptomatic. But this doesn't all matter in terms of transmission because, as I say, people do not transmit it to another person. Every human infection is a brand new spillover from a mosquito to a human. Very interesting, right? However, 1 in 150 people who get infected get neuroinvasive disease. The virus invades the central nervous system. You can get headaches, you have eye issues, muscle weakness, cognitive impairment, even paralysis, polio-like paralysis. This is a serious infection. 10% of the people who get neuroinvasive disease die, and over half of them have long-term neurological sequelae, cognitive issues, motor issues, difficulty moving. As I said, you do not want to have viruses multiplying in your brain. It's never a good thing. So here's a summary of the pathogenesis, a couple of things I want to point out to you. So we have a mosquito, right, involved in a cycle with birds, transmitting virus back and forth. Occasionally it will bite a human. The virus, of course, is delivered to the blood. It's picked up by dendritic cells. It's brought to the lymph node where it reproduces in lymphocytes. Viremia gets established. The virus spreads to other lymphoid organs like the spleen where there are lots of lymphocytes, so amplification. And that is, in most cases, what happens. In a fraction of cases, the virus gets into the brain. So these cells here are the blood-brain barrier. And it's interesting that the virus is sensed by toll-like receptor 3, and that sensing, of course, turns on the production of cytokines, one of which is tumor necrosis factor alpha, TNF alpha. And that's meant to be protective, but TNF alpha disrupts the endothelial barrier in the brain so the virus can get across. And that's why you have neuroinvasion in a certain fraction of people, because TLR3 sensing the virus, making a lot of TNF, maybe a lot in some patients. Virus gets in the brain where it can infect lymphocytes and will also infect neurons and cause the neurological sequelae. So that's how the virus gets into the brain by an unfortunate side effect of TLR3 sensing the virus and making TNF. It's immunopathology, basically. Here are some cases in the U.S. to give you a sense of the numbers. In different years, you have different numbers of cases. So you can see 1999, of course, that's the year it came in. And then it starts spreading across the U.S. We have big outbreaks in 2002 and 3, and then smaller ones here, and then bigger ones again. And we're not quite sure what determines the number of cases every year. One idea is that it's climate. When there's a particularly dry year, perhaps the birds are not available for the mosquitoes to bite, so they bite humans instead. On this graph, you can see the total number of cases every year and the number of deaths at the bottom. So not huge numbers, but if this is your mother or father, you're not happy about that. So we would like to have a vaccine for people who are at risk, that is, people who are outside and in contact with mosquitoes. And on the top, these are the neuroinvasive cases. So many thousands of neuroinvasive cases where these people have long-term sequelae. So that's not a good thing. So a vaccine is in development. Just a few slides on numbers in the U.S. This is the average annual incidence from 99 to 2019. And the darker states have higher incidence. So you can see Midwestern largely. We have some here in the Northeast, but not a lot. States where there's high density of these mosquito populations. It's seasonal. You can see it's peaking in the summer months, which is when the mosquitoes are active, and we're outside and we're able to be bitten by mosquitoes. The mosquitoes are not active in the other months, of course. And the incidence of neuroinvasive disease goes up with age. So it's very low in young people, and it goes up slowly with age until it's very high, incidence per 100,000, 1.2 in people over 70. So obviously age is doing something. We don't know if it's allowing higher virus titer replication, if it's causing more permeability, higher TNF alpha production. We simply don't know. But like many other virus diseases, it does increase with age. So right now we don't have a vaccine, although there are horse West Nile vaccines available. Why is that? Because horses are expensive and people don't want to lose their horses. So we made a West Nile vaccine for them. It's 30% fatal in horses. So there is a horse vaccine, but it's not approved for human use. But it might be at some point, in which case, if you're at risk for mosquito bites, then you would use it. So if you're living in Maine, I guess in the summer you would have some risk. But if you remember the chart, there wasn't a lot of West Nile in Maine. But at the moment, well, first of all, the culicine species that transmit the infection, the virus, they bite from the evening to the morning. They don't bite during the day. So if you're going to go out at those hours, you simply use repellents, you make sure you have screens on your house, and you wear the right clothing, which is unfortunately boring because you're supposed to wear long-sleeve shirts and you tuck your pants into your socks. You look like a complete nerd if you do that. But that's the way to prevent it. You can't wear shorts, for example. So until we have a vaccine, that's the story. I want to make one last comment about viruses in the central nervous system. We've talked today about several that invade the CNS: polio, measles virus, West Nile virus. They are all transmitted in other ways, right? So the gut, fecal, oral, respiratory tract, mosquitoes, that's how the viruses are transmitted. Going in the CNS is a dead end for a virus. So it's an accident. There's no evolutionary selection for the ability of a virus to get in the CNS directly because it does not contribute to transmission. As we'll see when we talk about evolution, the biggest selection pressure for a virus is to transmit well. And going in the CNS simply doesn't do that. And so next time we're going to talk about a very different kind of infection that lasts the life of the host, the persistent infection.